Hello, I'm Laura Cassidy from the American Chemical Society. Welcome to this news briefing from ACS Spring 2021. We're joined today by Dr. Tobias Widener from Aarhus University. He's studying how snake species from different terrains surrender surface secrets behind their slithering process. Dr. Widener?
Thank you very much, Laura. So there's my slides. Yeah, thank you very much for the opportunity to present our snake research here. So so basically, people have always been fascinated with the sliding and slithering locomotion of snakes, and also engineers and material scientists have been quite fascinated with the low friction and high durability properties of snake skin, and also of the ability of snakes to negotiate difficult terrain and like sandy slopes or climb trees and so forth. And so my collaborator Stanislav Gorb from Kiel University, who's also an author on this presentation that we're going to have this later today. He is interested in the biomechanics of the snakes, and he's been looking at this snake skin scale with microscopy, and as you can see on the slide here, so there's the scales on the belly side, or on the back side, so dorsal and the ventral side. When you look at the microstructure, they're actually quite similar. So there you can see this on the right hand microscopy images here. But then the belly of this, in this case, this is a California king snake, and it's very low friction on the belly where it slides on, and it's very high friction on the back. And the question was, why are these friction properties so different if the microstructure is so similar? And so we did a study where we basically came in. So my group at Aarhus University, we are specializing in looking at surface chemistry, and we do spectroscopy at surface to really see the outermost 1, 2, or 3 layers of molecules of materials. And so we collaborated with Stas Gorb to look at what is defining the chemistry of snake scales. And Laura, you can maybe show the next slide. And basically, we found on this California king snake, to our surprise, that there's a nanometer thin layer of lipids sitting on top of the scales. And this was also a big surprise for biologists, because basically the skin structure of snakes is pretty well known. And so we found this layer of lipids on top. And the interesting thing was that we found that the lipids were very ordered on the belly of the snake, and ordered lipid layers make very good lubricant. And on the back, there were lipids, but they were not very well ordered. So that has a higher friction. And so basically the snake has a lubricating layer, a nanometer thin layer, where it needs it. And so this Mette Rasmussen, a PhD student who's now working on this project, so basically she's going to give a talk about this later today. And she looked into, is this just coincidence? Is this snake just happen to have this layer? Or is this something that is maybe correlated really with the habitat? And so on the next slide, you see 3 different snakes that Mette has been looking at. And so it's a ground snake, a sand snake, and a tree snake. And basically she found that on the ground snake, which is the California king snake, and the tree snake, again, there are highly ordered lipids on the belly and disordered lipids on the back. And the sand snake, which is very different from the other two, because it really dives through the sand and really lives sort of in the middle of it. So as you see here, it's like it's coming up as the head, but most of the time it's really sliding through the sand. And this snake has ordered lipids all around, so on the belly and on the back. And so it looks like that these snakes are really using these lipid layers as a friction reduction mechanism, and is really correlated with their lifestyle or with their habitat. And yes, and that's of course also could be very interesting for engineers who try to mimic snake skin for robotics, or engineer low friction materials, to really not only look into the microstructure, which is typically done, but also look at what is really the outermost surface chemistry that defines the material and its contact with its environment. Thank you very much. That's all I have.
All right. Thank you very much. Are there any questions from viewers? Please state your name and affiliation in the chat window when you ask your questions. While we're waiting for questions from viewers, I have a few of my own. First of all, what got you interested in studying snakes?
So that was mostly due to, so I had a collaboration with Woodland Park Zoo in Seattle. That was when I was still a postdoc at the University of Washington. And yes, and so basically I was starting with an insect entomologist there, and it turned out that we had a project where we tried to understand the chemistry, the surface chemistry of spider. It turned out spider feet are quite rough. It was quite difficult for us, and obviously snake skin is more flat, and so it was a very good starting point for these kinds of studies, because it's easier to do spectroscopic studies. Yeah.
Okay. In what types of settings might it be useful for robots to mimic snake locomotion?
So typically when material scientists or engineers, when they look for inspiration in biology for making structures, very often they focus very much on the structure. So what is the morphology of a surface? Is it rough? Is it flat? And so forth. And I think people are very good at this. But then I think our results can maybe be used to add another viewpoint to these studies, and that is, what is really the chemistry of it? What is the chemistry that sort of like codes these structures, for example, that you also find on snakes and that are very well known? And maybe compared with like making a microstructure, it could actually comparatively easy to have a chemical coating of a surface. For example, a lipid coating, or for example, is probably easier to make than a sophisticated microstructure. So it could be quite easy to make some progress there.
Good. All right. Why hadn't people previously noticed that snakes are covered in a lipid layer?
Well, it's a very, very thin layer. It's just a few nanometers of lipids. And then when you look at lipids consist of basically hydrocarbons and some other elements. So very similar to whatever is below the surface they're sitting on. And so chemically. And so it's very easy to overlook them, because you really need some really very surface sensitive techniques that can look at really the outermost few nanometers. And I guess also when you study snake skin, they have this very rich microstructure, and I think there's lots of study there. So people were not immediately looking for other sort of like other aspects of this.
Okay. Are the lipids found on snake scales different from those on other slimy reptiles?
I mean, first of all, so basically, I think typically snakes are not really considered to be slimy. So it's actually something that people tell you for people who are afraid of snakes or don't want to touch them, then biologists typically tell them you can touch them. They are not slimy. They're actually dry to the touch. And then basically our study shows that that's not entirely true, because they are a little bit slimy, but only on the nanoscale, right? So they're only nano slimy, if you want. But I don't know lipids on slimy reptiles. I think the surface, but I'm really not an expert on this, but I think there's some sort of mucus that's chemically also different from a lipid layer.
I see. Okay. Do you think that this oily layer is secreted by the snake's skin cells? And have skin cells been shown to do this in any other species?
So I'm not aware of a study on snakes. I'm sure that otherwise skin scales are very good at transporting lipids and oily types of substances. So it's something we are looking into. We're trying to find out how can snake skin move lipids through these layers and to the surface, because we're interested to find out, are the snakes picking the lipids up in their environment, or are they producing them themselves and then somehow transporting them to the scale surface? But we don't know.
Okay.
But I would assume that there are lipids in the scales, since it's also important for them to be. They would be very dry and sort of brittle normally if there's no lipids and no oily substances. But I don't know that, so that's something we're trying to find out.
Mm-hmm. Where did the snake skins come from in your study?
They come from our collaborator, who's also an author on the talk that Mette will be giving this later today, Stas Gorp at Kiel University. So they have a facility where they keep the snakes, or used to keep them. Yeah.
Mm-hmm. And I understand you studied recently shed skins. Is it possible that their surface chemistry differs from skin that's actually on the snake?
And that, I mean, we cannot exclude it, but the nice thing is, so when snakes shed their skin, then they really have sort of the surface chemist in mind when they do it, because they're basically turning the skin inside out. So at least, of course, that's not, I mean, we cannot fully exclude it, but at least when you find the shed skin in the cage or whatever, where they're kept, then they're sort of turned inside out, and the inner the surface is sort of protected. Yes, that's
Okay. Have you studied when in the snake's life cycle this lipid layer appears? For example, have you studied the skins of baby snakes?
So the snakes we've studied are all adult snakes. We don't know when it appears. Yeah.
Okay. Have you looked at the skin of snakes that glide across water yet?
We would very much like to do this, because I think when you go from a solid surface, dry surface, to a liquid surface, especially water, then of course the surface chemistry that you would want differs very much. So I mean, so I would be just interested, is it a hydrophobic surface? Is it hydrophilic? Does it maybe like, for example, in shark skin, you have a combination of a micro or nano structure and then also a sort of a coating on top. I mean, do we find something like this? So I would be very interested. The difficulty is to make the samples, because apparently the skin of water snakes also decays very quickly somehow. So it's a different chemistry, and of course it's more difficult to make a meaningful sample. So we have to sort of make a very good have a good design, so we can be sure that what we're looking at is really the relevant surface chemistry.
Okay. And can you give us a little more detail on how you determine the orientation of the lipid molecules on the snake scales?
Yeah, so basically, we use two types of spectroscopies to do this. One is a vibrational spectroscopy that is very surface specific. It's called some frequency generation spectroscopy, and there basically you can basically see the difference of different molecules in these lipids when they are sort of vibrating in one direction or they're vibrating in the other direction, if you will. So you can look at the pointing direction. And then we're using a electron spectroscopy. Basically, we shine in X-rays, and then we get electrons out, and we can analyze those, and then find out what side is up and which side is down.
Okay. How might a snake keep the lipids denser and highly organized in some parts of its surface, but less dense and less organized in other parts?
I mean, this goes a little bit back to the question: how are they actually producing this layer? Is there something in the skin that makes this? Of course, if you have typical lipids, if you have only few lipids on a surface, then they tend to lie flat on the surface, or they're crumbling up. And then if you have more lipids, then they come together and make a densely packed layer, because they like to be on the surface. And then you have more of them, then they kind of stand up and pack really well. So I guess it's more a matter of how many lipids, like what's the amount of lipids on the surface. And if it's really a mechanism that's in the scales, then it will be easy to explain. Then it could be that the scales on the back on a ground snake just transport fewer lipids.
Okay. Now, when you compared the lipid layer between snake species, were there differences in the thickness of the layer in addition to the differences in the lipid orientation?
so we cannot say this from our data. We can only say sort of the layer is thinner than about 5 nanometers in all cases where we see the lipids, but it's hard for us to say exactly, do we have, is it 2 nanometers or 5 nanometers? That's hard to tell from the data.
Okay. In our press release on your work, you mentioned that the organization and density of the lipid layer are more important than its chemical makeup. Can you explain that statement in more detail?
Yeah, so basically for the friction properties of the lipid layer, it's more important. So that basically, typically what you have in a lipid is you have an alkyl chain, and then you have some sort of functional group. And so it's a polar molecule. And then sort of the friction properties come more from the alkyl chain. And the differences there are not so, sometimes you have slight differences or different branchings, but looking from a distance, it's sort of similar. So they all, when they pack really well, provide good lubrication. And the head group is probably more important for anchoring the lipids to the skin surface, or anchoring them to a second layer of lipids. So in this sense, I'm not saying the head groups are unimportant, but I think for the lubrication part, it's not so important.
I see. Okay. Do you have an educated guess as to which types of lipids might make up this layer?
No, I don't.
That's fine.
Yeah.
Okay.
I mean, we would love to work with, if somebody hears this, who is doing a lipodomics, for example, we would be very interested to collaborate, and we could provide samples. I think could be very interesting to see, especially for different species. So we would have a nice library of samples, of course, but I would be interested in it, but I don't know.
Mm-hmm. That is okay. So for people who are developing snake-like robots, how might they apply your findings?
Well, they could, depending on what type of robot they want to make, they could apply a coating, a lubricating lipid or oil coating to their robot. I mean, in a way, is like lubricating something with an oily substance is not a completely new concept, I guess. So that's something they could do. But in general, I would say maybe what I hope is that that maybe there's that people start looking more at the chemistry of these surfaces, the chemical identity of it, because there's a lot of research going into looking at understanding the structure. So like, what is the shape of it? Is it rough or not, flat? And there's less research going into the chemistry. And I think in some cases there could be some interesting inspirations we could take from nature.
Sure. And what are your next steps for this research?
Well, with this, I mean, basically you already asked about it. So I think I would be very interested to look into the water snakes, to see if there's really, because there could be quite a dramatic difference in the coating. And then, of course, we also what we are interested in also extending this to other species, and see what is the role of surface chemistry in those, in animals that are controlling their hydration. They're basically the animals that cannot be wet, or others that are very good at staying hydrated. There are many animals that are sort of clinging to surfaces really well, and others are non-stick. And we're very interested in exploring the role of surface chemistries in these processes.
Okay, my final question today is, what take-home message about this research do you want to leave with viewers?
I don't know, look at you should judge these functional surfaces from the surface chemistry. So it's quite important when you have a surface like of an animal. The outermost few nanometers really define its interaction with the environment, and so I think it's quite exciting to look at that, because oftentimes we find very surprising adaptations and really interesting tricks of nature.
All right, thank you very much, Dr. Widener. The archived version of this session will soon be posted at www.acs.org/ACS Spring 2021 Conferences. Please join us for our next press conference tomorrow at 2 p.m. Eastern. Thank you.